Welcome to the Deep Dive, your shortcut to being well-informed. We take complex topics, sift through the details, and bring you the clearest, most important insights. Today, we're plunging into a really foundational subject, the immune system. Our mission, well, it's to distill Chapter 18 from Vander's Human Physiology, breaking down its intricate mechanisms, key players, and core concepts. Think of this as getting the essential understanding, like a last-minute lecture from an expert, so you walk away truly well-versed in how your body defends itself.
That's exactly right. We're focusing exclusively on this chapter's material, explaining technical terms in clear language and following its structure to build a complete picture of this incredibly vital system. We're here to help you connect the dots.
Okay, great. Let's unpack this amazing defense network your body has, starting with the big picture.
Yeah.
At its core, what is the immune system? It's not quite like a single organ system, is it?
No, not at all. It's far more diffuse than that. Unlike, say, the digestive system with its connected organs. The immune system is more of a dispersed collection of disease-fighting cells. You find them throughout your body, in your blood, lymph, various tissues and organs. It's essentially your body's way of telling self from non-self, foreign stuff, and then dealing with anything that's foreign.
Right. So what's its primary mission then? What are these scattered cells actually trying to achieve?
Well, it really has three core functions. First, and maybe the most obvious one, it protects against infection from pathogens, think viruses, bacteria, fungi, parasites.
Yeah.
Second, it isolates or removes any foreign substances, and that includes both living microbes and non-living particles. And third, and this is really critical, it destroys cancer cells. That process is called immune surveillance.
Okay, three main jobs. And the chapter immediately introduces two big sort of overarching strategies the immune system uses, and they're constantly interacting, right?
Exactly. We classify immunity into innate and adaptive responses. Innate immunity, you can think of it as your body's initial general defense force. It reacts to foreign substances without needing to recognize their specific identities. Its mechanisms aren't really tailored to one particular invader, so we often call them non-specific immune responses.
Okay, the general first line, and the other side of that coin, the more specialized approach.
That would be the adaptive immune response. This is more like your immune system's special operations unit, if you will. It relies on specific recognition by cells called lymphocytes of the exact substance or cell that needs to be attacked. So these are also known as specific immune responses. And what's fascinating is how these two arms work together in innate immunity can actually provide sort of instructions to activate and guide the adaptive responses.
Interesting how they cooperate. Before we dive into the cells themselves, let's quickly touch on the main adversaries. The chapter focuses on two primary types of pathogens.
Yes, primarily bacteria and viruses. They're the dominant infectious agents, especially in industrialized nations. Bacteria are single-celled organisms. They can damage tissues directly or sometimes release harmful toxins. Viruses, on the other hand, they aren't really even considered living organisms. They're basically just genetic material wrapped in a protein coat, and they have to hijack your own cells to multiply.
Ah, so they use our machinery.
Exactly. And that process often kills those host cells. Think of things like the flu virus or SARS-CoV-2, prime examples of viruses doing their thing.
Got it. Now, let's meet the key players, the cells and the chemical messengers that make all this happen.
Okay, the main cellular components are your leukocytes, or white blood cells. Now, unlike red blood cells, which pretty much stay in your bloodstream, leukocytes are mobile. They can actually leave the circulatory system and enter tissues, which is where a lot of the immune action takes place. These cells broadly fall into two main groups based on where they originate from stem cells.
Okay, let's start with the first group, the myeloid cells. Who are the stars in that category?
Right, the myeloid cells, these are largely your immediate responders and the cleanup crew. Key players include neutrophils. These are really abundant phagocytes.
Phagocytes meaning they eat things.
Yes, exactly. They engulf and destroy foreign particles, and they also release inflammatory chemicals. Then you have monocytes. These circulate in the blood, but quickly move into tissues where they transform into these larger, more potent phagocytes called macrophages. Macrophages are found in almost every organ, especially at interfaces like your skin or the lining of your digestive tract. Their main job is phagocytosis, just devouring particulate matter. They also present antigens, which is crucial for signaling other immune cells.
Okay, so neutrophils and macrophages are big eaters. Who else in the myeloid group?
There are also dendritic cells. They're similar to macrophages in their phagocytic ability, very motile, found in areas exposed to the environment. They're critical for processing pathogens and then migrating to activate other immune cells. And finally, mast cells. You find these in connective tissues. They're packed with these little vesicles, these sacs, that release chemicals like histamine, which really kicks off those immediate innate immune responses.
Okay, so those are the generalists, the first responders. What about the specialized, more targeted cells, the lymphoid group?
Ah, yes, the lymphoid cells, these are your lymphocytes, the special operations force, you could say. First, B lymphocytes or B cells.
Yep.
These are responsible for initiating antibody-mediated responses. When they get activated, they mature into plasma cells, which are basically just antibody factories,
Antibody factories, okay. And the T cells.
right? The T lymphocytes are key cells. These mature in an organ called the thymus and are involved in what we call cell-mediated immunity. And there are a few really critical types here. You've got cytotoxic T cells. These directly seek out and destroy specific target cells, like cells infected with viruses, cancer cells, or even transplanted cells that are recognized as foreign. Then there are helper T cells. These are absolutely crucial coordinators. They don't attack directly, but they secrete chemical messengers called cytokines that activate pretty much everyone else. B cells, other T cells, NK cells, macrophages. The whole system really struggles without them.
Or linchpins, it sounds like.
Very much so. And then there are regulatory T cells. These act as a vital brake on the immune system, suppressing other immune cells to prevent things from getting out of hand or attacking the wrong targets. And one more group, natural killer cells, or NK cells. These can bind directly and non-specifically to virus-infected cells and cancer cells and kill them. They provide a really important immediate defense while the more specific adaptive responses are gearing up.
Wow, that's quite a diverse team of cells. How do they all communicate and coordinate their efforts effectively?
Yeah, it's complex. That's where cytokines come in. These are essentially small protein messengers. They're secreted by immune cells and actually some non-immune cells too, and they regulate how cells divide and function. They're the chemical communication network, basically. They allow immune cells to talk to each other. We call that crosstalk.
And these cytokines, they have many different jobs, right? It's not just one signal.
Oh, absolutely. Most act locally, influencing nearby cells, but some can travel through the bloodstream like hormones and cause widespread effects. They often create cascades, where one cytokine triggers the release of others, amplifying the whole response. Key examples are cytokines that kickstart inflammation and fever, like interleukin-1 or TNF-alpha. Others stimulate immune cell growth and activity, like interleukin-2, and some even help fight off viruses directly, like interferons. They really orchestrate the entire immune response.
Okay, fascinating. Now, let's zero in on that first line of defense. Innate immunity. How does your body respond right away without needing specific prior knowledge of the invader?
Right. Innate responses are all about recognizing general molecular features, things that are common to many invaders, like specific carbohydrates or lipids you'd find on microbial cell walls. It's not about recognizing the unique identity of one specific bug, which is the key difference from adaptive responses.
So what are the absolute first barriers a pathogen would encounter?
Well, your body surfaces are critical. Just having intact skin is a huge barrier. Then you have things like the hairs in your nose, the cough or sneeze reflex. These are all physical hurdles. And then there are chemical defenses too. Glands secrete antimicrobial chemicals like lysozyme that actually destroys bacterial cell walls, and lactoferrin, which binds up iron, basically starving bacteria that need it. Plus, you've got sticky mucus that traps particles, which are then swept away by cilia or maybe swallowed. Even your stomach acid is a potent killer for many ingested pathogens.
Okay, physical and chemical walls. But then things get more active, right? Here's where it gets really interesting. Inflammation. We all recognize the signs redness, swelling, but what exactly is happening there physiologically?
Yeah, inflammation. It's the local response to any infection or injury. Its job is really twofold. First, destroy or inactivate the invaders, and second, prepare the area for tissue repair. The main phagocytes involved here are those neutrophils, macrophages, and dendritic cells we mentioned. And those familiar signs you mentioned, local redness, swelling, heat, and pain, they're all driven by chemical mediators released at the site.
So what are those mediators actually doing to cause those specific signs like redness and swelling?
Okay, so mediators like histamine, they trigger vasodilation. That means widening the small blood vessels and also increased permeability, making the walls of the capillaries and venules leakier. This increased blood flow causes the redness and warmth, and it allows protective proteins and leukocytes to flood into the area from the blood. That increased leakiness, the permeability, also causes plasma fluid to filter out into the surrounding tissue. That leads to edema, which is the swelling.
And how do those immune cells, the leukocytes, actually get from the blood to the site of action?
Through a really neat process called chemotaxis. Circulating neutrophils, for example, first start to sort of roll along the inside surface of the blood vessel that's called margination. Then they actively squeeze through the endothelial cells lining the vessel wall that's a diapetesis. And they do this by following a chemical trail, like breadcrumbs, laid down by chemoattractants like chemokines, leading them right towards the pathogens. Monocytes follow a bit later, transforming into macrophages once they arrive, ready to join the fight and clean up.
Okay, they follow the trail. Once they arrive, how do these phagocytes, the neutrophils and macrophages, actually destroy the pathogens?
They destroy them mainly by phagocytosis, essentially, eating them. Their surface receptors bind to those general pathogen features like carbohydrates or lipids. Sometimes this binding is enhanced by chemical factors called opsonins. The word literally means to prepare for eating. They basically make the pathogen more tasty or recognizable to the phagocyte.
Like putting a flag on it.
Exactly.
Ah,
Once engulfed, the microbe is trapped inside the phagocyte in a little sac called a phagosome. This phagosome then fuses with lysosomes, which are like the cell's recycling centers, full of enzymes creating a phagolysosome. Inside that potent chamber, enzymes and other destructive chemicals like nitric oxide and hydrogen peroxide just break down the microbe.
wow, quite the process. The chapter also highlights the complement system as another crucial part of innate immunity. How does that work? It sounds complex.
It is a bit complex, yeah. The complement system is a family of about 30 plasma proteins that normally circulate in an inactive state, kind of like a set of dormant grenades. But when they get activated by an infection or cell damage, they unleash a cascade reaction. A key step involves activating a protein called C3. When C3 gets activated, one piece, called C3b, can get deposited directly onto a microbe's surface. This C3b acts as a really potent opsonin again, flagging that pathogen for destruction by phagocytes.
Okay, so it flags things. Does it do anything else?
Oh, yes. C3d also triggers the formation of something called the membrane attack complex, or MAC. This structure literally embeds itself into the pathogen's plasma membrane, forming these leaky, pore-like channels. This disrupts the pathogen's internal environment and essentially kills it.
That sounds pretty dramatic. And how does this complement system get activated in innate immunity without the specific antibodies that we associate with adaptive immunity?
Good question. There's an alternative complement pathway. It basically bypasses the need for antibodies entirely. It gets triggered directly by interactions between certain microbial surface molecules, like carbohydrates, and some of the inactive complement proteins. This provides a rapid, antibody-independent way to get that C3b onto the invader.
Okay, so there are ways to activate it quickly. We've got inflammation, phagocytes, complement, physical barriers. What's another key innate defense the chapter mentions?
Right, interferons. These are a type of cytokine, and they're grouped into type I and type 2. Type I interferons are produced by most cell types when they get infected by a virus. They then bind to receptors on both infected and nearby uninfected cells. This triggers those cells to synthesize antiviral proteins that generally interfere with viral replication. So it's like warning the neighbors to lock their doors.
A general alarm system against viruses.
Exactly. And these actions are nonspecific, meaning they work against many different kinds of viruses. Some viruses, like SARS-CoV-2 actually, have evolved ways to try and limit this interferon production. Type 2 interferon, which is also called interferon gamma, is produced more by immune cells like NK cells. It primarily helps boost the bacteria-killing activity of macrophages and acts as a chemoattractant during inflammation.
Okay. Now, stepping back a bit, how do these innate immune cells, the macrophages, dendritic cells, actually recognize these general foreign features you mentioned, these PMPs, without specific antibodies? What's the sensor?
Great question. They do this using special receptor proteins called toll-like receptors, or TLRs. These are found on cells like macrophages and dendritic cells. They belong to a larger family called pattern recognition receptors, PRRs. Think of them as generic security sensors. They are designed to recognize those pathogen-associated molecular patterns, PMEPs. We talked about these conserved molecular features that are vital to a pathogen survival, like a specific type of lipid or nucleic acid that's common to many bacteria but not found in our cells.
So they detect these fundamental microbial patterns.
Precisely. When a PMP binds to a TLR on, say, a macrophage, it generates signals inside that cell. This leads to the secretion of inflammatory cytokines and the activation of other immune cells. These TLRs represent a very ancient, very fundamental way our bodies first evolved to sense microbial danger.
Really fundamental stuff. Okay, let's transition now to the other major arm, the highly specific adaptive immunity. Where does this incredibly precise defense system begin?
It all starts with the lymphocytes, those B cells and T cells we introduced earlier. They have this incredible ability to recognize specific foreign material. And any molecule that can trigger such an adaptive immune response is called an antigen. Most antigens are proteins or large polysaccharides. You find them on the surface of viruses, bacteria, cancer cells, or even transplanted cells. It's the lymphocyte's ability to distinguish between millions of different antigens that provides the incredible specificity of this adaptive response.
Okay, specificity is key. What are the three core stages the chapter describes for how a typical adaptive response unfolds?
Right, it follows a general pattern. First is encounter and recognition. Each lymphocyte, during its development, creates a unique receptor that's designed for just one specific antigen. When it finally bumps into that antigen and binds to it, it's considered recognized. Each lymphocyte is specific for only one type of antigen. Second stage is lymphocyte activation. That antigen binding acts like an on switch, activating the lymphocyte. This leads to rapid cell division, a process called clonal expansion, creating a whole army of identical daughter cells, all specific for that same antigen. Some of these daughter cells become effector lymphocytes. These are the cells that actually carry out the attack. Others become memory cells, which hang around for a long time, ready to provide faster, stronger immunity if you encounter that same antigen again later.
Clonal expansion, like making photocopies of the right soldier.
Exactly. And the third stage is the attack. The effector lymphocytes launch their assault. Activated B cells, remember, differentiate into plasma cells that churn out specific antibodies to target the pathogens. Activated cytotoxic T cells directly find and kill any cells bearing the specific antigen they recognize. After the threat is neutralized, most of those effector cells actually die off through a process called apoptosis, programmed cell death. But those crucial memory cells persist, sometimes for years.
And where do these specialized lymphocytes, B cells, and T cells actually live and get organized? Where does all this activation happen?
Good question. Lymphocytes predominantly reside in specialized lymphoid organs. We categorize these into primary and secondary. The primary lymphoid organs are the bone marrow, which is where B cells mature, and the thymus gland, located in your chest, where T cells mature. These organs supply the body with a stream of mature but still naive lymphocytes, meaning they haven't encountered their specific antigen yet. Then you have the secondary lymphoid organs. These include your lymph nodes, those little bumps you can sometimes feel, the spleen, the tonsils, and also clusters of lymphocytes found in the linings of your gut, respiratory tract, genital and urinary tracts, basically, anywhere pathogens might try to enter.
So the secondary organs are where the action happens.
That's right. These secondary lymphoid organs are the crucial meeting places, the battlegrounds, where those naive lymphocytes actually encounter antigens presented by other cells and get activated. And importantly, lymphocytes don't just sit still. They're constantly recirculating, moving between these secondary organs, the lymph fluid, the blood, and back into tissues. This constant traffic greatly increases the chance that a rare lymphocyte will eventually find its specific antigen.
Okay, constant surveillance. Let's quickly differentiate again between the B cells and T cells, specifically in the context of these adaptive responses.
Sure, B cells, once they're activated by their specific antigen, and usually with help from helper T cells, differentiate into those plasma cells. And plasma cells are dedicated to secreting large quantities of antibodies specific for that antigen. This whole process is known as an antibody-mediated or humoral response. It's particularly effective against extracellular threats like bacteria floating in your body fluids, viruses before they get inside cells, and toxins. T cells, on the other hand, are the drivers of cell-mediated responses. We have the cytotoxic T cells, often called CD8 plus cells, which, as we said, directly kill infected cells, cancer cells, or form graft cells. They have to physically travel to their targets to do this. Then the crucial helper T cells, or CD4 plus cells, they don't kill directly, but they help by secreting those vital cytokines that amplify and direct both the B cell responses and the activity of cytotoxic T cells and macrophages. And finally, the regulatory T cells, also usually CD4 plus 8, act as that essential brake, suppressing other immune cells to prevent autoimmunity or excessive damage.
Okay, clear distinction. Now, how do these cells, particularly B and T cells, actually recognize their specific targets at the molecular level? What do these receptors physically look like?
Both B cells and T cells have highly specific receptor proteins embedded in their plasma membranes. For B cells, the receptor is actually a membrane-bound version of the specific antibody, or immunoglobulin, Ig, that its progeny, the plasma cells, will eventually secrete in large amounts. An immunoglobulin molecule is typically Y-shaped, built from 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains linked together.
Right, that classic Y shape. So the structure must be really important for how it works.
Absolutely. It's a perfect example of structure dictating function in physiology. The stem of the Y, called the Fc portion, is relatively constant within a given class of antibody, like IgG or IgM. This Fc part is important because it can interact with receptors on other immune cells, like phagocytes, and also with the complement system. But the tips of the 2 prongs of the Y, these contain the antigen binding sites. These are the highly variable regions. The amino acid sequences here vary immensely from one B cell clone to another. This variation creates millions upon millions of unique antigen binding sites, each shaped like a specific lock, designed to bind perfectly to only one specific antigen key.
A lock and key mechanism.
Exactly. And B cell receptors can bind to antigens that are free-floating, or antigens still attached to the surface of a foreign cell, like a bacterium.
That's incredible diversity. How can the body possibly create millions of different antigen binding sites, millions of different keys from a relatively limited number of genes, maybe only about 200 or so genes involved?
Yeah, it's a fantastic biological puzzle. This immense diversity arises from a unique genetic process that happens only in developing lymphocytes. Basically, the DNA segments that code for these antigen binding site regions are physically cut, randomly rearranged or shuffled, and then rejoined together. This cutting, shuffling, and rejoining process varies randomly from B cell to B cell as they develop. It's like shuffling a deck of genetic cards in millions of different ways, creating an almost limitless repertoire of unique B cell receptors and therefore antibodies.
Wow, genetic shuffling. Okay, what about T cell receptors? Are they similar?
T cell receptors are structurally related to antibodies. They're also part of the immunoglobulin superfamily and have variable regions that bind antigens. They also achieve their diversity through similar DNA rearrangement processes, but this happens in the thymus where T cells mature. But there are 2 huge differences. First, T cell receptors remain embedded in the T cell membrane. They are not secreted like antibodies. Second, and this is absolutely critical for understanding T cell function, a T cell receptor cannot bind to a free antigen like a B cell receptor can.
It can't. So how does it recognize the antigen?
A T cell receptor can only recognize and bind to an antigen when that antigen is first presented to it, complexed with certain of the body's own plasma membrane proteins.
Ah, okay. So the antigen has to be sort of held out for the T cell by one of our own cells, and these self-presenter proteins have a special name.
Exactly. These crucial self-proteins are called the major histocompatibility complex, MHC proteins. In humans, you'll also hear them called human leukocyte antigens, HLAs. Think of them as cellular identity tags or display platforms on the cell surface. What's really interesting is that your set of MHC proteins is unique to you, like a molecular fingerprint. No 2 people, except identical twins, have the exact same set of MHC proteins. This MHC diversity is why tissue transplantation can be so tricky.
Okay, unique identity tags. Are there different types?
Yes, there are 2 main classes. Class I MHC proteins are found on the surface of virtually all nucleated cells in your body, basically everything except red blood cells. Class II MHC proteins, however, are found primarily on the surface of specific immune cells, particularly macrophages, B cells, and dendritic cells, the cells we often call professional antigen-presenting cells. And this distinction is vital for T cell function. Cytotoxic T cells, the CD8+ ones, generally recognize antigens presented by class I MHC proteins. Helper T cells, the CD4+ ones, generally recognize antigens presented by class II MHC proteins.
So the type of MHC determines which type of T cell can see the antigen, and this whole process is called antigen presentation.
Precisely. Antigen presentation is the process where cells display antigen fragments bound to MHC molecules on their surface for T cells to inspect. And the cells that do this are called antigen-presenting cells, APCs. Let's look at how it works for helper T cells first. Remember, they need class II MHC. So only macrophages, B cells, and dendritic cells typically act as APCs for them. Imagine a macrophage engulfs a bacterium, phagocytosis. Inside, it breaks the bacterium down into small peptide fragments. These are the actual antigenic determinants or epitopes. These fragments then get loaded onto class II MHC molecules, and the whole complex MHC plus antigen fragment is transported to the macrophage's surface. There, it's displayed waiting for a specific helper T cell whose receptor happens to fit that particular MHC antigen complex. For full activation, the helper T cell usually needs a second signal too, a co-stimulus, often involving interaction between other matching proteins on the APC and T cell surfaces, plus cytokines like IL-1 released by the APC.
Okay, so that's for helper T cells using class II MHC from specialized APCs. What about presenting antigens to cytotoxic T cells? They use class I MHC, right?
Correct. And since almost all nucleated cells have class I MHC, potentially any infected cell or cancerous cell in your body can act as an APC for a cytotoxic T cell. The antigens presented by class I MHC are typically endogenous antigens, meaning they originated inside the cell itself. Good examples are viral proteins produced inside a virus-infected cell or abnormal proteins produced by a cancer cell. These endogenous proteins are broken down into fragments within the cell, loaded onto class I MHC molecules, usually in the endoplasmic reticulum, and then the complex is displayed on the cell surface. This basically signals, "Help, I'm infected," or "Help, I'm abnormal," waiting for a specific cytotoxic T cell to recognize the complex and kill the compromised cell.
That makes sense. Endogenous threats shown on class 1, exogenous threats processed and shown on class 2.
Broadly speaking, yes. That's a good way to think about it.
Now, with all this incredible machinery for recognizing foreign antigens and even distinguishing self-MHC from foreign MHC, how does the body normally avoid attacking its own healthy tissues? That seems like a massive potential problem.
It's a critical question, and the answer lies in immune tolerance, the fundamental ability of your immune system to remain unresponsive to your own body's self-components. Your body works hard to establish and maintain this self-tolerance, mainly through two key mechanisms during lymphocyte development. First, clonal deletion. This happens primarily in the primary lymphoid organs, the thymus for T cells, the bone marrow for B cells. During their maturation, any developing leukocyte whose receptor happens to bind too strongly to a self-antigen presented there is essentially triggered to undergo apoptosis, programmed cell death. They are deleted before they can cause trouble. Second, clonal inactivation or anergy. Some potentially self-reactive lymphocytes might escape deletion and make it out into the periphery. However, if they encounter their self-antigen out there without the proper co-stimulatory signals, which are usually only provided by activated APCs during a real infection, they often become inactivated or non-responsive. They're still there, but they're effectively muzzled. It's a multi-layered safety system.
Okay, deletion and inactivation as safety checks. Let's try to put this all together now. Can we trace an actual adaptive immune response, say, against a bacterial infection, step by step?
Absolutely. Let's walk through a typical scenario. 1. Bacteria manage to get past the initial innate defenses and enter the body, maybe eventually reaching a lymph node or the spleen. 2. Inside the lymph node, a naive B cell whose surface immunoglobulin receptor happens to perfectly match a surface antigen on one of those bacteria will bind to it. This is the initial recognition step for the B cell. 3. Meanwhile, a macrophage, or dendritic cell, in the same area engulfs some of the bacteria through phagocytosis. It processes the bacterial proteins into peptide fragments and presents these fragments on its surface bound to class II MHC molecules. 4. This APC now subjects for a naive helper T cell, whose T cell receptor specifically recognizes that particular bacteria-derived peptide presented on the class II MHC. When they connect, the APC provides the necessary co-stimulatory signals and releases cytokines like IL-1 and TNF-alpha, fully activating that specific helper T cell.
Okay, so the helper T cell is now activated. What does it do?
5. The activated helper T cell starts secreting its own powerful cytokine, interleukin-2, IL-2. IL-2 acts back on the helper T cell itself, causing it to proliferate rapidly, clonal expansion, creating many copies. It also releases other cytokines. 6. Now, these activated helper T cells can find and interact with those B cells that have already bound the same bacterial antigen. The B cell internalizes some antigen and presents it on its own class II MHC. The helper T cell provides crucial help to the B cell, both through direct cell-to-cell contact and by releasing specific cytokines like IL-2 and others. This interaction fully activates the B cell.
So the B cell needs the T cell help to get fully going.
Often, yes, especially for protein antigens. This T cell help is crucial for a strong, effective antibody response. 7. Once fully activated, the B cell undergoes its own massive clonal expansion and differentiation. Most daughter cells become plasma cells, those antibody factories we talked about, dedicated to pumping out large amounts of antibodies specific for the original bacterial antigen. A smaller number become long-lived memory B cells. 8. The secreted antibodies, mostly classes IgG and IgM in this kind of response, circulate through the blood and lymph, eventually reaching the site of infection. There they bind specifically to the bacteria they recognize.
And once the antibodies bind to the bacteria, how do they actually direct the attack? What do they do?
They orchestrate the destruction in several key ways. First, direct enhancement of phagocytosis. Antibodies act as opsonids. Their Fc stem region binds to Fc receptors on phagocytes, like macrophages and neutrophils, essentially forming a bridge between the phagocyte and the antibody-coated bacterium. This makes phagocytosis much more efficient. Second, activation of the classical complement pathway. When certain antibody types, IgG or IgM, bind to antigens on a bacterial surface, their Fc regions change shape slightly, allowing the first complement protein, C1, to bind. This kicks off the classical complement cascade, leading ultimately to the formation of that membrane attack complex, MAC, which can directly kill the bacteria, and also generating more C3b for even better opsonization. Third, antibody-dependent cellular cytotoxicity, ADCC. Antibodies can link target cells, like bacteria or even infected host cells, to natural killer NK cells. The antibody binds the target, and the NK cell binds the antibody's Fc region. This triggers the NK cell to release toxic chemicals that kill the target cell. It confers antigen specificity onto the NK cell's killing action. And fourth, direct neutralization. Antibodies can bind directly to bacterial toxins, preventing them from harming host cells. Or they can bind to surface proteins on viruses, preventing them from infecting host cells. This binding can also cause pathogens to clump together, making them easier targets for phagocytes.
Wow, antibodies are really versatile weapons then. Flagging, activating complement, linking to NK cells, neutralizing. Quite a toolkit.
Absolutely. They are central to clearing many types of infections.
And you mentioned memory cells, so this process explains how the body remembers previous infections and fights them off better the second time.
Exactly. That's the basis of active immunity. The very first time you encounter a specific pathogen, the primary response, it takes time to activate the right lymphocytes, undergo clonal expansion, and produce antibodies. It's relatively slow, and the antibody levels aren't huge. But thanks to the memory B cells and memory T cells generated during that primary response, if you encounter the same pathogen again later, a secondary response, these memory cells are already primed. They get activated much more quickly, proliferate much faster, and produce a much larger amount of antibody much more rapidly. The response is so fast and strong you might not even feel sick the second time.
And that's how vaccines work, right? They trigger that primary response safely.
Precisely. Vaccines use killed pathogens, weakened pathogens, or harmless components, like specific proteins or inactivated toxins, to induce active immunity and generate those memory cells without causing the actual disease. So your body is prepared for the real thing.
Okay, that's active immunity. Your body does the work. What about getting immunity without having to fight the infection yourself?
Right, that's passive immunity. This involves the direct transfer of preformed antibodies from one individual to another. The classic natural example is a mother transferring Ig antibodies across the placenta to her fetus, providing protection for the newborn for the first few months. Or IgA antibodies transferred through breast milk. Clinically, we can sometimes administer injections of pooled antibodies, gamma globulin, or specially engineered antibodies to provide immediate but temporary protection against certain toxins or infections. The protection is immediate because the antibodies are already there, but it's short-lived because the recipient's body didn't make them and will eventually clear them out, and no memory cells are formed.
Got it. Active is long-lasting memory. Passive is temporary protection. Now, let's shift focus slightly. How does the immune system defend against threats inside our own cells, like virus-infected cells or cancer cells? It's a different kind of challenge, isn't it? Attacking your own altered cells.
It is a different challenge, yes. The goal here isn't just to clear extracellular pathogens, but to identify and destroy host cells that have gone rogue, either because they're harboring viruses or because they become cancerous. The primary weapon for this is the cytotoxic T cell, CTL. Remember, virus-infected cells or cancer cells display those endogenous antigens, viral proteins or abnormal cancer proteins, complexed with their class I MHC molecules on their surface. Specific CTLs, whose T-cell receptors recognize these particular MHC antigen complexes, will bind to these target cells. However, for full activation and proliferation into an effective killing force, CTLs usually also need cytokine help, particularly IL-2, provided by activated helper T-cells.
So helper T cells are still crucial here, even though the killing is done by cytotoxic T cells.
Yes, absolutely. Helper T-cells get activated by APCs, like dendritic cells, that pick up free viral antigens or cancer antigens and present them on class II MHC. These activated helper T cells then release IL-2 and other cytokines. This IL-2 then stimulates the nearby antigen-specific CTLs to proliferate massively and become efficient killers. Once activated, the CTL travels to the target cell, binds tightly to the MHC antigen complex it recognizes, and then delivers a lethal hit. It releases proteins like perforin, which forms pores in the target cell membrane similar to the complement MAC, and enzymes called granzymes, which enter the target cell and trigger apoptosis programmed cell death. Essentially, the CTL tells the infected or cancerous cell to commit suicide. And just like with B cells, memory CTLs are also generated for long-term protection.
A very targeted execution. Are there other cells involved in killing these compromised host cells?
Yes. Natural killer NK cells also play a significant role. While their initial recognition can be nonspecific, they look for cells that are stressed or lack normal MHC-I expression. Their killing activity can be greatly enhanced by antibodies through that ADCC mechanism we discussed, or by cytokines like IL-2 and interferon gamma released from activated helper T cells. Additionally, activated macrophages whose killing power has been boosted by helper T cell cytokines like interferon gamma, can also destroy infected or cancerous cells, often by releasing toxic chemicals. So it's a team effort, but CTLs are the primary specific killers for cells presenting endogenous antigens on MHC-I.
Okay, a multi-pronged attack against internal threats too. Now, infections clearly affect more than just the local site of invasion. What are some of the widespread systemic effects your body experiences during an infection? The chapter calls it the acute phase response.
That's right. Infections don't just stay local, they trigger systemic responses throughout the body. This whole collection of responses is known as the acute phase response. And it's important to realize that many of these aren't just unpleasant symptoms of being sick, they are actually adaptive adjustments your body makes to fight the infection more effectively.
Interesting. So what are some key signs of this acute phase response?
Well, fever is a classic one. While uncomfortable, a moderate fever can actually enhance protective immune responses and inhibit the growth of some pathogens. You often see a decrease in plasma iron and zinc concentrations. This is thought to be adaptive because many bacteria require high levels of iron to multiply effectively, so hiding it away hinders them. Your liver dramatically increases its production of a variety of acute phase proteins. A well-known example is C-reactive protein, CRP, which can act as a non-specific opsonin, helping to clear pathogens and damaged cells. Others help minimize tissue damage, or aid repair. Your bone marrow ramps up production of leukocytes, especially neutrophils and monocytes, providing more troops for the fight. There are also significant metabolic changes. Amino acids might be released from muscle protein breakdown to provide building blocks for new immune proteins, and fatty acids released from adipose tissue to provide energy. And importantly, there's usually an increase in the secretion of cortisol, a stress hormone. While cortisol can suppress some immune functions long-term, in the short term, it's thought to help modulate the response, preventing it from becoming dangerously overactive, acting as a kind of negative feedback.
So it's a whole body mobilization. What's orchestrating all these diverse, widespread effects? How does the message get out?
These systemic responses are primarily orchestrated by those pro-inflammatory cytokines we keep mentioning, especially interleukin-1, IL-1, tumor necrosis factor alpha, TNF, and interleukin-6, IL-6. These are released mainly by activated macrophages and other cells at the site of infection or injury. But they don't just act locally. They enter the bloodstream and act like hormones, traveling to distant targets like the brain to induce fever, the liver to stimulate acute phase protein production, the bone marrow to boost leukocyte production, and muscle and fat tissue to alter metabolism. It's the immune system's way of putting the entire body on high alert and coordinating resources for the battle.
Makes sense. Now, your body's ability to fight off infection isn't always constant, is it? What kinds of factors can actually influence your resistance, make you more or less susceptible?
No, it's definitely not constant. Many factors can significantly alter your resistance to infection. Probably the single biggest factor worldwide is protein-calorie malnutrition. If you don't have adequate protein and energy, your body simply can't synthesize the essential immune proteins, antibodies, cytokines, receptors needed for an effective response. Pre-existing diseases can also play a major role. For example, diabetes mellitus can impair the function of leukocytes, making individuals more susceptible to infections. Any kind of tissue injury, even sterile injury, can lower local resistance in that area. And there's a huge connection between your stress levels and state of mind and your immune function. The immune system is intricately linked with the nervous and endocrine systems. Chronic stress, for instance, often suppresses immune responses, while acute stress might sometimes enhance certain aspects. It's complex.
What about lifestyle factors like exercise or sleep?
Yes, those matter too. Modest, regular physical exercise generally seems to have beneficial effects on the immune system and overall health. Conversely, sleep deprivation has been shown to reduce the activity of important cells like NK cells. And of course, certain drugs can decrease resistance, most notably the immunosuppressant drugs used to prevent rejection in transplant patients, which intentionally target and reduce leukocyte production or function. This leaves patients more vulnerable to infections.
Okay, a lot of factors. One of the most significant and tragic examples of compromised immunity is, of course, AIDS. What exactly is happening immunologically in AIDS?
Right. Acquired immune deficiency syndrome,
All
AIDS,
right.
is caused by the human immunodeficiency virus, HIV. HIV is a retrovirus, and it has a devastatingly specific target. It preferentially infects helper T cells. It does this because a protein on the helper T cell surface, the CD4 protein, which we mentioned helps helper T cells recognize class II MHC, along with another core receptor, unfortunately acts as the primary receptor for HIV to enter the cell.
So HIV targets the very cells that are supposed to coordinate the entire adaptive immune response. That sounds catastrophic.
It is. That's exactly why it's so devastating. HIV replication inside helper T cells directly kills them, but it also triggers the body's own cytotoxic T cells to attack and kill infected helper T cells. And there's evidence HIV can even cause apoptosis, programmed cell death, in uninfected helper T cells through indirect mechanisms. So, over time, typically years after the initial infection, the number of functional helper T cells in the body gradually declines. Once the count drops below a critical level, the entire adaptive immune system essentially collapses. B cells don't get the help they need to make effective antibodies, and cytotoxic T cells don't get the help they need for activation and proliferation. This leaves the body profoundly vulnerable to a whole range of opportunistic infections. Infections caused by microbes that wouldn't normally harm a healthy person, and certain types of cancers, like Kaposi's sarcoma, that are normally kept in check by the immune system. These are the defining characteristics of AIDS, along with symptoms like weight loss and fever, often driven by high cytokine levels.
How is HIV/AIDS managed now? Is there a cure?
There's no cure yet, unfortunately, but therapeutic management has improved dramatically. Treatment typically involves combinations of powerful antiviral drugs, often referred to as HAART, highly active antiretroviral therapy. These drugs work by inhibiting key viral enzymes like reverse transcriptase, which the retrovirus needs to copy its RNA into DNA, or protease, which it needs to assemble new virus particles, or by blocking the virus from entering cells in the first place. These drug cocktails can effectively suppress viral replication, slow down the destruction of helper T cells, and significantly delay the progression to AIDS, allowing people with HIV to live much longer, healthier lives. But they don't eliminate the virus completely, and lifelong treatment is usually required. Developing an effective vaccine remains a major scientific challenge due to the virus's high mutation rate and its ability to hide within cells. Prevention remains key, avoiding transmission through contaminated blood, using protection during sex, and preventing mother-to-child transmission.
Okay, a difficult challenge. What about bacterial infections? We rely heavily on antibiotics there, but that comes with its own set of problems, doesn't it?
Yes, antibiotics have been revolutionary for treating bacterial infections. These are substances originally derived from microorganisms themselves that selectively kill bacteria or inhibit their growth without harming human cells. They typically target processes unique to bacteria, like building their cell walls, which our cells don't have, making their specific types of proteins, or replicating their DNA.
But we hear a lot about concerns with antibiotic use.
We do, and for good reason. First, some people can have allergic reactions or experience toxic side effects. But the biggest long-term concern is the development of antibiotic resistance. Bacteria reproduce incredibly quickly, and random mutations can occur. If a mutation happens to make a bacterium resistant to an antibiotic and that antibiotic is present, then only the resistant bacteria will survive and multiply, eventually leading to a population that the drug can no longer kill. Bacteria can also directly transfer resistance genes to each other on small pieces of DNA called plasmids.
So overuse or misuse of antibiotics drives resistance.
Exactly. The more we use an antibiotic, the more selective pressure we put on bacteria to evolve resistance. Indiscriminate use, like taking antibiotics for viral infections where they have no effect, or not finishing a full course of treatment, really accelerates this problem. Another issue is that broad-spectrum antibiotics can wipe out not just the harmful bacteria, but also the beneficial bacteria that normally live in our gut and elsewhere, our microbiota. These good bacteria usually help keep potentially more dangerous ones in check, so eliminating them can sometimes create an opportunity for harmful microbes to overgrow, leading to secondary infection.
Right, a complex balance. So the immune system is usually our protector, but we've hinted that sometimes its own actions can actually cause harm or disease. Can we explore some of those situations?
Absolutely. It's a critical aspect. The immune system is powerful, and when it's misdirected or overreacts, it can definitely cause problems. A major example is graft rejection. When tissues or organs are transplanted from one person to another, a graft, the recipient's immune system almost always recognizes the donor tissue is foreign. Why? Because of those MHC proteins we discussed. Unless the donor is an identical twin, their MHC molecules will be different from the recipient's. The recipient's T cells, particularly cytotoxic T cells, recognizing foreign class IMHC, and helper T cells, recognizing foreign class II MHC presented by APC's processing donor cells, mount an attack against the perceived foreign cells, eventually leading to the destruction and rejection of the graft.
So how do we manage transplants then?
We have to use immunosuppressant drugs. Medications like cyclosporine, which blocks the production of IL-2, a key cytokine for T-cell activation, or corticosteroids, which have broad anti-inflammatory and immunosuppressive effects, are used to dampen the recipient's immune response enough to allow the graft to survive. But this is a balancing act. These drugs suppress the entire immune system, not just the part attacking the graft. This leaves the transplant patient much more susceptible to infections and even certain types of cancer. Plus, the drugs themselves can have significant side effects.
A delicate balance indeed. What's another example of harmful immune responses?
Transfusion reactions. This happens when red blood cells are destroyed during a blood transfusion because of incompatible blood types. Red blood cells don't have MHC proteins, but they do have other surface molecules, primarily proteins and carbohydrates, that can act as antigens. The most well-known are the ABO blood group antigens.
Right. So if a person with type A blood who has A antigens on their red cells and anti-B antibodies in their plasma accidentally receives type B blood with B antigens, their anti-B antibodies attack the transfused type B cells.
Precisely. Most people naturally have pre-existing antibodies, called natural antibodies, in their plasma against the ABO antigens they don't have on their own cells. So giving someone incompatible blood triggers a rapid and potentially severe destruction of the transfused red blood cells. This is why cross-matching blood types carefully before transfusion is absolutely critical. Another important red blood cell antigen system is the Rh system, particularly the Rhd antigen, often just called the Rh factor. Unlike ABL, people who are Rh negative don't usually have pre-existing anti-H antibodies. They only develop them after being exposed to Rh positive red blood cells. This becomes clinically important in hemolytic disease of the newborn. If an Rh negative mother carries an Rh positive fetus, some fetal red blood cells might leak into her circulation, especially during delivery. This sensitizes her, causing her to produce anti-Rh antibodies. If she later carries another Rh positive fetus, her anti-Rh antibodies, which are typically IgG and can cross the placenta, can enter the fetal circulation and attack the fetus's red blood cells, causing potentially severe anemia. Fortunately, we now have ways to prevent this sensitization in Rh negative mothers.
Okay, rejection and transfusion reactions make sense. What about allergies? Those seem like the immune system overreacting to harmless things.
Exactly. Allergies are a type of hypersensitivity, an immune response that's exaggerated or inappropriate, directed against normally harmless environmental antigens called allergens, leading to excessive inflammation and tissue damage. There are different types, but the most common one we call allergy is immediate hypersensitivity, which is also known as IgE-mediated hypersensitivity. Here's how it works. The first time a susceptible person is exposed to an allergen, like pollen, dust mites, certain foods, they don't have a reaction then, but they become sensitized. Specific helper T cells stimulate B cells to produce an unusual class of antibody called IgE, specific for that allergen. This allergen-specific IgE doesn't just circulate. It binds very strongly via its Fc stem to the surface of mast cells found in tissues and basophils in the blood. These cells are now armed with IgE. Now, upon re-exposure to the same allergen, the allergen molecules bind to and cross-link these IgE antibodies sitting on the surface of the armed mast cells. This cross-linking triggers the mast cell to rapidly degranulate, releasing a flood of potent inflammatory mediators, most famously histamine, but also leukotrienes, prostaglandins, and cytokines. These mediators then cause the rapid symptoms of allergy. Vasodilation, increased vascular permeability, leading to swelling, runny nose, smooth muscle contraction in airways, causing wheezing, itching, and mucus secretion. This happens very quickly, hence immediate hypersensitivity. Common examples are hay fever, hives, and allergic asthma.
And sometimes these reactions can be really severe, right? Anaphylaxis?
Yes. If the mast cell degranulation happens systemically throughout the body, often due to an allergen entering the bloodstream, like from a bee sting or certain foods in highly allergic individuals, it can cause anaphylaxis. This is a potentially life-threatening whole-body reaction characterized by a massive release of mediators causing widespread vasodilation, leading to a dangerous drop in blood pressure or shock, airway constriction, making breathing difficult, and swelling. It requires immediate medical treatment, often with epinephrine.
Scary stuff. Okay, so allergies are attacks on harmless external things. What happens when the immune system loses that self-tolerance we talked about and mistakenly turns its powerful attack against the body's own tissues?
That's the basis of autoimmune disease. It's an inappropriate immune attack where the body's own self-proteins or other molecules act as antigens, triggering an immune response against the very tissues they belong to. This attack can be mediated by autoantibodies, antibodies that bind to self-antigens, or by self-reactive T cells, both helper and cytotoxic T cells. There are many different autoimmune diseases depending on which tissues or organs are targeted. Examples include multiple sclerosis, attack on myelin sheaths in the nervous system, rheumatoid arthritis, attack on joint linings, type 1 diabetes mellitus, attack on insulin-producing beta cells in the pancreas, and Graves' disease. Antibodies stimulate the thyroid gland.
What might cause this breakdown of immune tolerance? Why does the body start attacking itself?
That's the million-dollar question, and the causes are often complex and not fully understood. It likely involves a combination of genetic predisposition and environmental triggers. Some possibilities include a failure of those tolerance mechanisms like clonal deletion or inactivation, allowing self-reactive lymphocytes to survive and become activated. Alteration of the body's own proteins, maybe by infection or chemical exposure, so they look foreign to the immune system. Exposure of normally hidden self-antigens. Sometimes tissue damage might release proteins that the immune system hasn't normally encountered, and it mistakenly mounts a response. Molecular mimicry. This is where components of an infectious microbe might happen to look very similar, structurally, to a self-protein. An immune response targeting the microbe might then accidentally cross-react with a similar self-protein. A classic example of a systemic autoimmune disease is systemic lupus erythematosus, SLE, or just lupus. In SLE, the immune system produces autoantibodies against a wide range of self-molecules, particularly components of the cell nucleus like DNA and nuclear proteins. These antibody-antigen complexes, called immune complexes, deposit in various tissues, skin, joints, kidneys, blood vessels, triggering widespread inflammation and damage. Sunlight exposure can often trigger flare-ups in SLE.
So it's a failure of self-recognition in various ways. Finally, the chapter mentions that even appropriate inflammatory responses can sometimes become harmful if they're just too excessive.
Yes, absolutely. Inflammation is essential for fighting infection and healing, but too much of a good thing can be damaging. We saw this dramatically in cases of septic shock. This can occur during severe systemic bacterial infections, sepsis, when macrophages release massive amounts of potent cytokines like IL-1 and TNS into the bloodstream. These cytokines, in such huge quantities, cause widespread vasodilation leading to severe hypotension, dangerously low blood pressure, leaky capillaries, high fevers, and often organ failure. It's the immune system's own overreaction that becomes life-threatening. We also saw similar issues with excessive inflammation, sometimes called a cytokine storm, contributing significantly to the lung damage and severe disease in some patients with COVID-19. And beyond these acute situations, chronic, low-grade inflammation driven by persistent immune activation is now recognized as playing a major role in the development or progression of many chronic diseases, including things like asthma, rheumatoid arthritis, which is also autoimmune, inflammatory bowel disease, and even atherosclerosis, hardening of the arteries.
So it really hammers home the point. The mediators of inflammation and immunity are truly a double-edged sword, essential for normal resistance and health when produced in the right amounts, at the right time, in the right place, but capable of causing significant illness and damage when they're generated in excess or directed inappropriately. It's all about maintaining that delicate balance.
That sums it up perfectly. Balance is key.
And that wraps up our deep dive into the immune system, a truly fascinating, incredibly complex, and absolutely vital network that diligently protects your body every single day.
We've covered a lot of ground today, from the fundamental distinctions between innate and adaptive immunity, the diverse roles of specialized cells like B cells, T cells, macrophages, and NK cells, through to the molecular signals of cytokines and complement, and that crucial process of antigen presentation via MHC molecules. We also explored how these powerful defenses are normally kept in check by immune tolerance, and what happens when things go wrong, leading to harmful responses like graft rejection, allergies, autoimmune diseases, and excessive inflammation.
Understanding these intricate mechanisms, even at this level, is really key to appreciating the incredible resilience of your body, but also the delicate balance required to maintain health. We really hope this deep dive has given you a clearer picture of this intricate system and maybe even spark some new questions about how this ancient and powerful defense system evolved to protect us and how it keeps learning throughout our lives.
Thank you for joining us for this in-depth look at Chapter 18.
From all of us on the Deep Dive team, thank you for learning with us. Until next time, keep exploring.